the lab, which aims to understand the neurobasis of and the behavioral effects of these techniques. As a side note, so I have been a professor at the Brain Institute for since 2008. Now I hold a position of a full professor in cognitive neuroscience. And as a side note, I have been practicing yoga, and this is part of my interest for this topic since 2003. I can say that I have a regular practice of yoga, and I follow a group in northern India, the Shivananda, which is my main line of practice of yoga. So in this study, what we tried to do was to advance a little bit more in the science behind a specific type of pranayama that I'm going to define it a little bit later, which is called Bhastrika pranayama. And the idea here was to investigate the impact of 30 days of this Bhastrika pranayama practice in naive individuals. So in other words, people that has never practiced yoga or pranayama before. We decided to then investigate changes, particularly changes in the mood field. So we investigated both behavioral and the neural changes before compared to after 30 days of practice of Bhastrika pranayama, in behavioral aspects of anxiety, behavioral aspects of affect, and on brain functional activity and connectivity using a randomized controlled trial. Just to give you a little bit of background, I hope I'm not, as we say in Portuguese, raining on the wet. So the science, the current science behind pranayama is a lot focused on changes that are really significant on our autonomic nervous system. So it's very clear that the practice of pranayama leads to changes both in the sympathetic nervous system, autonomic nervous system, as well as in the parasympathetic autonomic nervous system. What it is known is that the changes on sympathetic or parasympathetic autonomic nervous systems depend a lot on the frequency in which the breathing is taking place. So what is, I wouldn't say pretty much standardized, but the current idea behind these practices is that slow breathing practices, they move to a increased sympathetic predominance. So you have a reduced heart rate and decrease in blood pressure. And on the other side, for fast breathing practices, you have the opposite, or reduces sympathetic predominance and increase parasympathetic predominance, which is the reduce and the low frequency component of cardiac variability and increased heart rate. There are very little information on the changes that occurs in the cognitive aspects after the practice of pranayama, and this was the main focus then of this study, especially because there are evidence, although very preliminary and not a lot of evidence, suggesting that the practice of pranayama goes beyond the autonomic nervous system and affects the central nervous system. And it affects the central nervous system in many different ways. So we do have already evidence that it affects different cognitive functions, such as attention, working memory, and perception, such as visual and auditory perception. There had been a few evidence showing that there are changes in mood and reduced stress, based mainly on one publication by Sharma et al. In 2014. So with this in mind, we decided to conduct these randomized placebo-controlled trial, evaluating 4 weeks of the practice of daily practice of Bhastrika pranayama, 5 days a week practice of Bhastrika pranayama, on traits and state of anxiety using the STAI inventory, which is State Trait Anxiety Inventory. We also used, to evaluate affect, we used the positive and negative affect scale, or simply PANAS-P or PANAS-N for positive and negative. We also used fMRI data in 2 different protocols that I'm going to explain in a little bit more detail further down the line, which involved a emotional regulation task and a resting state task, both conducted in a typical clinical fMRI scanner or clinical MRI scanner, a 1.5 Tesla scanner, in our university hospital, which is where our laboratory is located. And we were also interested in understanding eventual brain changes that we might find, to trying to find correlations between the behavioral changes and the functional MRI changes. To do so, we selected 30 in the, oh, sorry. So we selected 30 healthy young individuals, and we divided these 30 individuals in 2 groups. Half of the individuals were in the control group with control tasks, and half of the individuals were practicing the Bhastrika pranayama. A few of the exclusion criteria involved heart problem or chronic rhinitis, in other words, people that had obstruction in their nostrils, regular drug use that might interfere with the autonomic nervous system or cardiac autonomic modulation, any diagnosis or history of neurological and psychiatric disorders, and any contraindication to perform an MRI scan, such as metal in the body, pregnancy, and claustrophobia, and so on. This then was a randomized controlled trial. So as I said before, half of the individuals were in one arm, which is the control group, and half of the individuals were in the pranayama arm or the active group. The control group, they also had an activity. So they would come to the lab and perform ludic activities with board games. So they stayed in the lab for 30 minutes playing different ludic games. And the pranayama group, they came to the lab to practice the pranayama. So that you have a better idea of how this was performed, the individuals in both groups, they came to the lab 3 times a week. Monday, Wednesday, and Friday. And they were asked to practice at home on Tuesdays and Thursdays. So the idea here was that in both groups, people were practicing for 30 days, 5 days a week, in being 3 days in our lab in both groups, and 2 days at home. And they also kept a diary of the days that they were capable of performing the tasks at home and the days that they were not able to do it. This is our sample for this study. So it's pretty much well balanced between the both groups. We didn't find any social demographic or clinical characteristic that differentiate both groups in baseline. So in terms of gender, in terms of age, in terms of education, in terms of household income, in terms of minimum wage, in terms of the baseline anxiety trait and state, the baseline positive and negative affect. So for all these characteristics, both groups were matched. With these 2 groups, then we divided, as I said, in a group of the practice of control. So the control group would play different games. They would come to the lab 3 times a day and play some games. And then 2 days at home, they would keep on playing these games for 30 minutes. The Bhastrika pranayama, it was defined based on the Swami Kuvayalananda. And the idea here is that this type of Bhastrika pranayama was composed of a Kapalabhati followed by the Surya Bedana. So the idea here is in the Kapalabhati, they practiced in order to achieve a 30 repetitions of the Kapalabhati, which involves an active expiration. So in the country of how we usually breathe, in which we have a active inspiration, in this type of in Kapalabhati, you have an active expiration and a passive inspiration. So they were trained to achieve 30 repetition of the Kapalabhati. So they would do the 30 repetition of the Kapalabhati. This was followed by a Surya Bedana and a series of bandhas. So they have an active inspiration through the right nostril. And then they have an apnea with the three bandhas, the Jaladhara bandha, the Uddiyana bandha, and the Bula bandha. And after the bandha, in a comfortable apnea period, they would go through a left nostril aspiration. These practice, the Kapalabhati followed by the Surya Bedana and the bandhas and apnea, was repeated for 25 minutes. So this study ran for 4 weeks, but as the practice of Bhastrika pranayama is very challenging, especially for people that didn't have any previous practice of pranayama, in the first week, they'd come to the lab for 5 days, from Monday to Friday. And in these 5 days, we had a very well-structured series of practices in order to guide them to a correct performance of the Bhastrika pranayama. So it was an increased difficulty in the level of the classes from Monday to Friday. And on Friday, we would guarantee that the individuals would be performing fairly well these practice of this type of Bhastrika pranayama. So Kapalabhati 30, and then followed by 1 repetition of the Surya Bedana, in which we also include the 3 bandhas, the apnea followed by the 3 bandhas. This is the timeline of the study. So the 30 individuals, 15 were in the pranayama group, the other 15 were in the control group. At baseline, we had the application of state and trait, anxiety and affect scales, the STAI and the PANAS, and a first fMRI acquisition, which was composed of a emotional regulation task and a resting state task. Then both groups would practice for 4 weeks. 3 times a week, they would come to the lab. 2 times a week, they would practice at home for 30 minutes. 5 minutes of introductory asanas and 2 minutes at the end with Shavasana. And in the middle, the 25 minutes of the Bhastrika pranayama or the 25 minutes or the control activities. After the 4 weeks, the individuals would come back to the MRI and repeat both the scales, STAI and the PANAS scale, as well as the fMRI, both the emotional regulation task and the resting state task. This, as I said, was performed by both groups in the same order as it is shown in this current slide. These are the behavioral changes and the way that we analyze the behavioral changes. So we had 2 scales, the STAI, which is capable of measuring state and traits, and the PANAS, both negative and positive, which is also capable of accessing state and trait. For each one of these variables, we performed a 2-way repeated measure ANOVA with 2 factors. The first factor is intervention, so whether they were in the pranayama group or the control group, and the other factor is time, so the baseline assessments and the assessments performed 4 weeks after the intervention. And we also used a post hoc test to do an intra-group analysis. And in this case, we used a student t-test to evaluate the intra-group changes, comparing before and after training for each group. These are the initial results we have for the effects of the pranayama in both groups. So what you see here in these two graphs are the state on the left and trait on the right. What you see in blue are the changes in the control group. What you see in red are the changes for the pranayama group. What you see in the y-axis are the scores for the STAI, and what you see in the x-axis is time. So baseline, before the intervention, and post training. What we found here is a significant interaction between groups. So the ANOVA revealed a significant change in the anxiety level of these individuals, showing that the control group was stable throughout the experiment. This I'm talking on the left for the state. So the state of anxiety didn't change for the control group, but it significantly reduced for the pranayama group, which you see there in red. These changes were revealed both by an interaction effect on the ANOVA, but as well as an intra-group variability that we found in the STAI for the pranayama group alone. So in other words, we did see a significant decrease in anxiety in the pranayama group as evaluated by the STAI scale. We also evaluated changes on the PANAS, or the positive and negative effect scale. What you see here then on the left are the PANAS, the changes in PANAS N. So in other words, PANAS N measures the negative effect. And what you see then on the two graphs on the left are the changes in the PANAS N, negative effect, for state, that's in the top, and trait, that's in the bottom. So please pay attention to these graphs on the left, and what you see is that there is a significant decrease in the negative effect on the pranayama group, and there is a relative increase in negative effect in the control group. This happens both for the state and trait, and what you see here then is a clear interaction detected by the ANOVA, a significant interaction, so showing that there is a significant effect of pranayama, which is independent of the control group, suggesting that there is a decrease in negative effect of the pranayama group. In the case of the positive effect, what you see is the pranayama seems to protect and do not decrease a positive effect, but it also doesn't increase positive effect. So as you see on the graphs on the right, and especially on the red line in the top right corner, what you see is the positive PANAS state, and then you see that there are no changes for the pranayama group, so it keeps stable, but in the control group there is a significant decrease in the positive effect. So these effects led to an interaction effect, an ANOVA interaction effect, also in the positive PANAS assessment, showing that there is a kind of a protection on the changes on positive effect for the pranayama group. As I mentioned, so these behavioral changes were also complemented by these methods. So the fMRI, which is a technique which is capable of detecting neuronal activity, brain activity in different brain areas. So both before and after training, all individuals in both groups, they participated in an fMRI protocol. So the fMRI protocol was conducted in a 1.5T scanner. in a university hospital. And while they were in the scanner, they had to perform 2 tasks. The first task, which I'm going to explain in a minute, is what we call the emotional regulation task or emotional regulation protocol. And this was followed by another protocol, which is the resting state protocol. Both protocols, the duration of the exams is on the order of 40 minutes. So each individual spent 40 minutes in the MRI scanner before training and after training. So it's very important to mention here that what we are looking is not for the brain changes that happened during the practice of the pranayama. What we are looking here are the brain changes that happened after the pranayama training. So 1 month after the pranayama changes, as compared to before the pranayama changes. The pranayama training, as compared to these changes that also might have happened in the control group. So this is the first task that the individuals had to do inside the MRI scanner before at baseline and 1 month after the training, and 1 month after the training, exactly. So when they were in the fMRI scanner, they would see images. So they could see both negative images, such as a car accident, which is the image I'm showing here, or a neutral image, or an image that doesn't have any kind of emotional impact when the individuals are looking to these images. And before they had access to the images, they were given an instruction. So the instruction was either look or reappraise. When they were asked to look to the image, they would just look at the image and try to understand the emotional impact of that image in the individual. If they were asked to reappraise, then their task was to turn that negative scene into a positive scene using this cognitive ability that we have to reappraise situations. So the idea here is, for instance, you see an image of someone in a hospital. When you were asked to look at this image, you just look at this image and appreciate the negative impact that these images have upon your emotional system. But when you're asked to reappraise, your task is to look at the image and, for instance, recreate that scene, thinking that that is just a movie, that's not real, that's not true, that's just an actor. When you do this reappraisal of the scene, you basically, what happens is that you also reappraise the impact that that image has on your emotional system. So after the individuals received the instruction, then they had an image. The instruction was either to look or reappraise when they received the image, which could be either negative or neutral. And then they would just try to reappraise or look at that image. Then they had a rating scale, a Likert 5 rating scale, in which they had to evaluate that image, saying if the image was very negative, neutral, positive, or very positive, and then a fixation cross. This was repeated many and many times so that we have the ability or the possibility to look for the brain areas that responded to either the emotional impact of the images, which happened during the look part of the protocol, but also look for the brain network involved in these more reappraisal parts of the protocol. We analyzed this data using 2 different strategies. So we first selected in the protocol the periods or the parts of the protocol that interest us the most. So we were interested in periods in which the individuals were just looking at negative images, which we called neg. or we looked at the changes in the brain when the individuals were trying to regulate the negative images or reappraise the negative images. And we analyzed this data using two strategies, both looking at the whole brain analysis with what we call small volume correction, or looking at specific regions of interest, which we call ROI analysis or region of interest analysis. The idea of the region of interest analysis is to understand that there is a simplified model of emotional regulation that involves a number of brain regions, including prefrontal cortex regions, such as the dorsolateral prefrontal cortex, the ventrolateral prefrontal cortex, the dorsomedial prefrontal cortex, the ventral medial prefrontal cortex, the anterior cingulate cortex, and the limbic system, including the amygdala. So with this in mind, what we did was to select these brain regions, so prefrontal cortices, insula, anterior cingulate, and amygdala. And what you see here are the regions of interest that we analyzed further for these individuals. What we found here is what you see in the top of these images. So what we find is that there are significant changes and mainly two regions here. So when the individuals were looking at negative images, they had a significant modulation in the amygdala, which is part of the limbic system and very important for emotional processing, and the left insula and the right insula, which is in a structure which is very important for emotional processing, especially disgust, but also is a system that's it's well implicated in the regulation of our autonomic nervous system, and also implicated in what we call interoception, which is the ability that we have to detect signals from our own body. What we see here in this sets of results is that there were significant changes in both groups, showing that there is an interaction effect in the amygdala and in the insula. In the bottom line, what we show are the correlations that we found between changes in brain activity and changes in the behavioral data, here coming from the PANAS, negative state and trait. So what you see here is that there is a significant correlation in the changes we found in the activity of the amygdala with the changes we found in the negative affect. This happened both in the amygdala as well as in the right and on the left insula. This was also the case for the reappraisal part of the protocol. So you see that in the reappraisal part of the protocol, there is also a modulation of the insula. So the insula seems to be involved both in the reappraisal as well as in the active evaluation of the images. But the reappraisal involved other areas, which are parts of our prefrontal cortex, such as this that we see, the left ventral medial prefrontal cortex, as well as the cingulate cortex, which you see in the middle. So for the reappraisal part of the protocol, we did find a very significant change in prefrontal cortex, in the anterior cingulate, and also in the insula. As for the resting state protocol, what the individuals had to do was, after they finished the emotional regulation protocol, they had to stay in the scanner with their eyes closed, lying down, try to be motionless, and not to interfere with whatever thoughts are arising in your mind. Data was continuously collected for 7 minutes, approximately 7 minutes, and the individuals were asked to pay attention to their own thoughts, their own emotion, that we were going to ask them to report what happened and what they thought during the acquisition of these resting state protocol. The idea then of the resting state protocol is to allow a further evaluation of the areas that changed activity by looking at how these areas communicate between each other. So in other words, we did find for the first part of the protocol that certain areas of the brain increased or decreased their activity after training as compared to before training. So pranayama was possibly associated with the pranayama change. The pranayama training was possibly implicated in changes in brain activity that happened mainly on prefrontal cortex, insula, and amygdala. Now we can ask a second question to our data, and we can ask it based on the resting state protocol, which is, so there is a change in activity. Now we want to ask, how is the communication between these areas that we found to be important for emotional regulation? So what we did here was to look for the connectivity, we called it functional connectivity, between these emotional regulation centers. Let me give you an example. So we looked for the insula and the amygdala, for instance. And then the question here is to understand whether the connectivity between these two brain areas increase or decrease after the pranayama change training as compared to the control change, to the control training. So the idea here is to look for the connectivity, so how different brain areas are connected between them, and what is the impact of the training, of the pranayama training, training in this connectivity between these two areas. This is what we find, and these are the results. So basically what you have here are the different brain, the different regions of interest. So for instance, here you see the right dorsal medial prefrontal cortex, the left dorsal medial prefrontal cortex. And what you see here as a dot is the location of that region. So the right ventrolateral prefrontal cortex, the left ventromedial prefrontal cortex, and so on, the orbital frontal cortex, the anterior cingulate cortex, and so on. So basically what we find is that there is a significant change in connectivity between the right ventral prefrontal cortex, ventrolateral prefrontal cortex, and the right ventrolateral prefrontal cortex. And these changes, they are, and we also found a correlation between changes in the anxiety with changes in the insula, both right and the left insula. So to wrap up what we have in this study, basically the results are pointing to the importance of pranayama in the modulation of three brain structures, mainly the prefrontal cortex, including ventral and lateral portions of the prefrontal cortex, and also the amygdala and the insula. These are the locations of these three main regions, which which is the main results of this study. So the main results are related to these three regions. And what do they mean? So the amygdala, so let's say the amygdala, the insula, and the prefrontal cortex. The amygdala is a structure that's part of the limbic system. And we know that the limbic system is a system that's very important for emotional processing, particularly negative emotional processing, such as fear. So there is a very strict relationship between the activity of the amygdala and fear. So for instance, when we are in the situation of stress, of fear, of anger, it tends to have to be followed by an increased activity of the amygdala. So what we found here is that with the pranayama practice, we found a change in the activity of the amygdala, suggesting that the emotional or the mood changes that we found are related somehow to these changes in the amygdala. This is well represented in other studies. So this is just an example of some of these studies in which they found that the mindfulness meditation practice is related to a decreased activity of the amygdala, suggesting that these practices, they have a profound impact on emotion and in emotional regulation, particularly in the processing of negative emotion. And the intuition we have here is that with the decreased activity of the amygdala, we are finding a decrease in these arousal of these emotional processing. What you see in the left is a increased activity of prefrontal cortices, also found in meditation practices. What does the insula has to do with meditation, and why do we think that the insula is a very important structure for that's being modulated by pranayama? This is a study by a Harvard professor, Professor Sara Lazar, that reported a structural change in the insula in experienced meditators. So what she found is that when you compare experienced meditators with naive meditators, there is an increased cortical thickness of this structure called insula. And the interpretation behind these change in the insula is that the insula modulate a cognitive ability that we have, which is called interoception. Interoception is the ability that we have to detect the internal signals of our body. So how aware are we to our own body? This is a construct that's called interoception. And it's very clear that the insula modulates interoception. So the insula is a substance that's, it's a brain structure that's very important to modulate interoception. So we believe that what pranayama does is the increased or the modulation of the activity we found and the connectivity we found in the insula is related to changes in interoception that these individuals felt with the practice of pranayama. So in other words, the practice of pranayama increases the individual ability to be aware to the internal signals of our own body. So the awareness to the body increases with the practice of pranayama, which seems to be the reason why we did see a change in the activity and the connectivity of the insula. And the third structure that's related to the changes we found is the prefrontal cortex. So the prefrontal cortex is this frontal regions in the brain, which is the biggest difference between humans and other species. The proportionality between the size of our prefrontal cortex with the rest of the brain is significantly higher than this proportionality in other animals. So the proportion between the prefrontal cortex and the rest of the brain in humans is a lot higher than the proportion in other animals. And the second aspect of the prefrontal cortex is that the prefrontal cortex is highly connected to other brain regions. The behavioral representation of the prefrontal cortex is fairly well understood. That's related to what we call in general cognitive control. So processes that are related to cognitive control. And this is pretty much what turns us into humans. So decision making, for instance, is part of cognitive control. Imagining the future is part of cognitive control. Imagining the past is part of cognitive control, and so on. Emotional regulation is part of cognitive
Let us wait for Professor because he has lost the connection. As he comes back, we will start this session again. Remaining.